Okay, so our first speaker this morning, like I said, will be Dr. Anil Shrestha. He's going to be giving us a talk on why do we see horseweed and hairy fleabane in most parts of California year-round in recent years. So Anil, whenever you're ready, go ahead and share your screen.
Some reason only I can't share my screen again. Okay, there I go. Can you see my screen now?
Yes, we can see it.
Okay. All right, so the title of my talk, as Whitney said, is why do we see horseweed and hairy fleabane year-round in most parts of the state in recent years. What I'm going to do is summarize some of my findings over the years of research with mostly these two Caniza species. So Caniza has been renamed to Erigeron, the genus name. So horseweed and hairy fleabane are both in this Asteraceae family, and they are technically classified as summer annuals in California, but we see their prevalence in orchards, vineyards, and you can name it, you can see it everywhere, including parking lots. So they can be pretty competitive in orchards, as you can see in this picture. This is horseweed, which can grow fairly tall, and then fleabane, which is about 2.5, 3 feet at the most, and you can see them in vineyards, orchards as well. Even on the plastic culture in the strawberry systems, you can see that these are all horseweed plants that are beneath the plastic. In the berry systems, in the annual crops, you primarily see them in field edges because they don't like disturbance, so they seem to prefer the field edges. But having said that, as you move to no-till systems, you can start seeing them in the field. This is from the Central Valley itself. It's no-till Roundup Ready cotton, and you can see these species there. In roadsides, it's fairly prevalent. Canal banks. So what I'm trying to get at is they have all kinds of habitats. Even in abandoned fields, you can see them all over the place, and vacant lots. And these two are pretty difficult to tell apart when they are at the seedling stage. The way you can tell the two apart is horseweed seems to be dark green in color, and the leaf is more of a club shape, whereas fleabane is lighter green in color, and it has a little bit of a serrated shape to it. But once it bolts, it's easy to tell the two apart. Fleabane also has less compact leaf. It's less leafier in general. And horseweed is more compact and has more leaves. It is a single stem, and whereas fleabane can branch. So often you find them growing together. This is horseweed, right here is fleabane. So these two, this is all based on what we've known about his biology like 10, 15 years ago. Both of them start flowering in July, but fleabane sets seeds about 2 weeks to a month earlier than horseweed, and they shed the seeds around July to October. And occasionally fleabane has been—
Horseweed and fleabane.
Pardon? Fleabane has been observed to, flower as early as March. So these are both very prolific seed producers. Horseweed, according to East Coast data, they produce up to 200,000 seeds per plant, but I have counted up to 800,000 seeds per plant under our conditions. Fleabane to 100,000 seeds per plant. This is what we believed was the life cycle of horseweed and hairy fleabane in Central California. We had 2 times of emergence. One was spring emergence and fall emergence. So the fall emergence would survive over the winter as a rosette and then start bolting and growing again the next season. Spring emergence would flower and set seeds by fall. So this is what we believed was the biology, and this is what we still see in publications. But what we're seeing is these seedlings are now, seen almost throughout the year. And that made me wonder, why is it that, we see it throughout the year in recent years, and what has changed in recent years? I think it's not just resistance. We have had resistance to Roundup in horseweed, and as well as fleabane, 8 to 16-fold resist 8-fold in horseweed, 16-fold in fleabane. But that's not just the whole story. It's just a part of the story of why we see it throughout the year. The main thing, I think, is the seed germination biology and ecology, how these things survive and germinate all kinds of stressful conditions. So when we look at plant physiology, for seed germination, we need optimum temperature, we need optimum light, we need moisture, and then pH, soil salinity, seed burial depth. These are more on the stressful side of things that kind of hinder germination. So I looked at all the data, like the studies that I have done personally and studies that other people have done. So the base temperature for horseweed germination, this is the minimum temperature that's required, is around 55 degrees Fahrenheit or 13 degrees Celsius. Fleabane can germinate earlier. In cooler temperatures, the base temperature is around 39, 40 degrees Fahrenheit or 4 degrees Celsius. So that's like the minimum temperature requirement. So we did a study to look at the effect of temperature on the germination. The optimum is around 77 degrees day and 68 nights of that would correspond to 25 Celsius, 20. But you can see that it can germinate at a range of temperature, and you just need a few plants for them to complete their life cycle. So that's the optimum, yes, but you can see that they can germinate in a range of temperatures, and we went only up to like 86, 77, but even at 40 degrees, I've seen germination. So if you look at the temperature, this is a Fresno area, we have the optimum temperature, or the minimum temperature, I should say, for germination of both these species almost throughout the year. And with warmer winters, we have the temperature that's required for them to germinate. So year round, they have the minimum temperature that's required for germination. So that's one reason why they can germinate. So that's one of the factors. Temperature is one of the factors. The other thing we've seen is that it needs light. It's not a species that will germinate without light. So you can see with the same temperatures, there's a drastic reduction in germination when there is no light. So it's what we call a photoblastic species. So it will need light to germinate. That's why it likes to germinate only from the surface of the soil, and anything buried deeper will not be conducive for its germination. The other thing we looked at is, okay, how tolerant is this to drought? You've seen this species growing in all sorts of areas where there's no irrigation at all. It's not very tolerant to drought for germination purposes. It does need a little bit of moisture, so it won't germinate in complete dry situation. Having said that, this is the back of my car. This is the trunk of my car. And I was doing some studies with these two species. I left the car in the garage over the weekend, and there's some water spill in the trunk, and that's all they needed. You can see they're germinating, but you can see they're not germinating on the dry side of the trunk. So, these can germinate anywhere as long as they have a little bit of moisture. And once they germinate and start growing, they grow like, crazy. They can tolerate drought to a high extreme. So this is not Mars, but parking lot kind of area at Fresno State, but they're growing like crazy. And they can complete their life cycle and produce viable seeds at all. Most of these seeds that you see are viable. And again, it beats the knowledge that we have about plant physiology on plant growth and development. So it has the temperature that it needs to germinate, but it needs a little bit of moisture to get started. Once it gets started, it'll grow like crazy. So the other stress factor for seed germination is pH. And we did a study to see the range of pH it can germinate from. We saw like, well, we tested from pH of 5 to 9, and we didn't see any drop in germination. We didn't test lower than 5 or higher than 9, because if you look at the state map, the soil pH is generally between the range of 5.5 to 8.8, around 8 in our state. So that it has the capability of adapting to most part of the states as far as soil's pH goes. Salinity, it's very tolerant to salinity during germination. So it can germinate at as high as 20 decisiemens per meter. So that's pretty dry, salt tolerant. And we have salt tolerant soils. I mean, not salt tolerant soils, but saline soils in many parts of the state. And you can see that 8 to 16 would be strongly saline, but these two species can germinate even at 20 decisiemens. So it can take over many of these saline areas as well. Now, the other question we had is, do these seeds of these species have to undergo a dormancy period? And we harvested the seeds and we tried to germinate it the very day we collected it, 7 days later, 14 days later, 21 days later. And you can see that they have the ability to germinate the day they fall off the mother plant. So they don't have to go through dormancy as long as there's moisture available. That's why you will see an adult plant growing, like this is fleabane, it's producing seeds. And you can see down here, there are seeds or seedlings that are already emerging, and those that have dropped from the mother plant. So this is a close-up of that. So we have a continuous life cycle throughout the year of these species. The temperature is there. As long as there's a little bit of moisture, it can germinate the day they fall off the mother plant. And most seeds fall and germinate close to the mother plant, but there are data showing that the seeds can travel hundreds of miles with the wind. So the other thing, as I mentioned earlier, is that they only germinate from the surface of the soil. Anything buried even 1/10 of an inch is not good for its germination. That's why we see it in no-till systems, undisturbed systems. Any sort of systems which has soil disturbance is not preferred by these species. So that is another reason, like we've been moving from all of these annual agronomic systems, vegetable systems, to more of the perennial systems, which are, these systems are high disturbance, so you won't really expect to see horseweed and fleabane as much until after the disturbance has been stopped. Here in a perennial systems, what they like, they just favor these species are adapted to these low disturbance systems. That's why we see them in more of a perennial system. Now, the nice thing about the seeds is they lose viability in about the 3rd, 4th year. They are not very, they don't last too long. So once they germinate and start growing, we wanted to know, well, they're germinating and growing and producing seeds year round, but do they have the growing degree days that is required for their completion of the life cycle? So this is a study where we were looking at the glyphosate susceptible and glyphosate resistant horseweed and tracking the growing degree days. So it requires about, like the susceptible one required more growing degree days, almost like 1600 something compared to 1400 something for the resistant ones. So they need at least 1400 to 1600 growing degree days for the completion of the life cycle. And the resistant one seems to set seeds about 25 days earlier than the susceptible one. But this is just one-on-one comparison with one particular population. You can't generalize this and say it will apply to all resistant and susceptible ones. Same thing with fleabane. They also require anywhere from 1300 to 1500 growing degree days to complete their life cycle. And if you look at our map, we have the growing degree days that is needed for them to complete their life cycle. So they can overwinter, they have rosettes that can easily tolerate frost and all that. So they have the temperature, they have the growing degree days, they have the light requirement that's needed to complete their life cycle. That's why, and they can germinate the day they fall off the mother plant. That is why they are seen throughout the year, I think. The other thing we noticed is the fall emerging ones require fewer growing degree days than the spring emerging ones to reach the rosette stage. Whereas in fleabane, it was the other way around. So there's subtle differences between these two conizers on how many growing degree days they require and what time of the year they germinate and emerge. So we've been modeling these in terms of growing degree days, and I've kind of looked at a model showing how many growing degree days does it require to get to the rosette stage, bolting stage, because most of the recommendations we have in terms of herbicide control is spraying by size or growth stage. I think that doesn't quite apply to this species or both of these species, because if you don't control them by 50 to 150 growing degree days in the rosette stage, so even though it's a rosette stage, what's happening is below ground is the story. They're producing longer roots and making it more difficult to control with herbicides. Once it gets to bolting stage, it's very difficult to control them. So for the post-emergence control window, it's about mid-April for spring populations, and for the fall populations, it's mid-November. And the same thing with fleabane. And you also have the post-emergence control around more mid-April and mid-November for the fall emerging. So the rosette stage, it can be deceiving as well. As I said, like it may look small, but it can be producing or growing below ground. And you can see this root system can be pretty deep. So that is how it survives drought conditions, I think. So a little bit about management issues. We've noticed that these two species are very sensitive to both temperature and light when it comes to being controlled by glyphosate or other herbicides. And we had better, even the resistant ones that we classified as resistant, we had better control when it was exposed to cold conditions than in the warm conditions. That's why I think that you get better control of these two species if you apply herbicides in fall, late fall, compared to spring. And I'm not sure why that is, but it's been reported in other places as well that there are conditions that make this happen. So if you're trying to control them with glyphosate, it's better to control them in fall than in spring when it's getting cooler. And we've been noticing it even with other types of weeds. And growth stage is important. So post-emergence herbicides. So again, although I said growth stage is important, as I mentioned, the rosette stage can be deceiving. So I'm almost thinking of, recommendations should be based on growing degree days. And once it gets to the rosette stage and beyond, your control just declines. This is data from Kurt Hembry and a little bit of studies that I was also involved in. And he's also found that the spray volume can also make a difference. And he recommended going with 40 to 50 gallons per acre for control of these species, both fleabane and horseweed. Otherwise, you would see regrowth. And also, he found that differences between the nozzles in terms of control. So this is turbo with 40 gallons per acre, turbo twin jet with 40 gallons per acre, turbo 50 gallons, turbo 50 twin jet 50 gallons. So he's recommending 40 to 50. And you can see here with glufosinate, 50 gallons per acre, you don't see much control, but 30. So minimum of 30 is what I would say for control of these species. And this is again data from Kurt. And when he compared the turbo and the twin jet, the regrowth from the twin jet was less. And he preferred that we use a twin jet for the control of these species. The other thing about hairy fleabane is it's not a very leafy plant, it's a very stemmy plant. So even with post-emergence herbicides, you got to have very good volume coverage on it to have success. Horseweed, on the other hand, is more leafier, so that's not the same as fleabane. And this is data just for fleabane. I don't think he did the same study for meadowtail or horseweed. So in conclusion, most parts of the state have temperatures that allow germination of horseweed and hairy fleabane year round. So as I showed you the map, the seeds, however, need a little bit of moisture to germinate, but once it germinates, it can tolerate very extreme drought stress, and it can grow and produce thousands of viable seeds. The seeds are very tolerant to pH and salinity stress for germination, so it can be adapted to any range of soil conditions for these two variables. But seeds do need light for germination, and they do not germinate from anything greater than 1/10 of an inch. And they do not like soil disturbance. So anywhere there's mulch or something like that, it can prevent germination of these species. And the seeds can germinate the day they fall off the mother plant, provided that there is available moisture, which makes a continuum of germination and emergence throughout the year. And they can mature, as I showed you, that they can mature, produce flowers and seeds throughout the year. So we have sufficient growing degree days that allow these two species to complete its life cycle year round. And they are pretty tolerant to frost. They can overwinter, pretty tolerant to heat, and still produce viable seeds. So what happens is, with these cropping systems, once we harvest the crop, we let go the weeds, and then the weeds are not done with their harvest process or their production processes. So they just keep producing seeds that add to the seed bank. And these seeds can disperse long distances. But as I said, if you do a good job in controlling the seed bank, I would say, the two strategies for weed production to reduce weed populations is to reduce reproduction or reduce dispersal, or not, or both. Both reduce reproduction and reduce dispersal. So seed bank management therefore becomes a very important aspect for any sort of weed control. And then conversion to perennial cropping system seems to be favoring them. And as I mentioned earlier, that they don't like disturbed systems. So they are not very prevalent in your agronomic systems, which have high disturbance. So efficacy of post-emergence herbicides seem to be, different in different seasons. So when I first started UC, with UC 20 years ago now, my first weed that I was exposed to was fleabane and horseweed. I've spent 20 years working with this weed, and there were millions of dollars, but not to me, but to the whole state, for different researchers to work on this species. We know how many genes it has, or what type of DNA it has, and everything there is to know about this, but they're still around. So sometimes it's humbling to work on weeds. You spend so much of your efforts, you spend your career, you spend dollars, but they're still there. So I don't know if we've solved any problem, at least not with conizers. So with that, I would like to thank you for your attention. And if there are any questions, I think with me, I may have a few minutes.